Don't stop. Don't stop. Don't stop. Don't stop. Don't.
Glands. We have seen that some epithelial cells may be specialized to perform a secretory function. Such cells, present singly or in groups, constitute glands. Unicellular glands are interspersed amongst other, non-secretory, epithelial cells. They can be found, for example, in the epithelium lining the intestines. Such glands develop as diverticulae from epithelial surfaces. The distal parts of the diverticulae develop into secretory elements, while the proximal parts form ducts through which secretions reach the epithelial surface. Those glands that pour their secretions onto an epithelial surface, directly or through ducts, are called exocrine glands, or externally secreting glands. Some glands lose all contact with the epithelial surface from which they develop. They pour their secretions into blood. Such glands are called endocrine glands, internally secreting glands. When all the secretory cells of an exocrine gland discharge into one duct, the gland is said to be a simple gland. Sometimes there are a number of groups of secretory cells, each group discharging into its own duct. These ducts unite to form larger ducts that ultimately drain onto an epithelial surface. Such a gland is said to be a compound gland. Both in simple and in compound glands, the secretory cells may be arranged in various ways. A. The secretory element may be tubular. The tube may be straight, coiled, or branched. B. The cells may form rounded sacs or acini. C. They may form flask-shaped structures called alveoli. However, it may be noted that the terms acini and alveoli are often used as if they were synonymous. Glands in which the secretory elements are greatly distended are called saccular glands. D. Combinations of the above may be present in a single gland. From what has been said above, it will be seen that an exocrine gland may be unicellular, simple tubular, simple alveolar, or acinar, compound tubular, compound alveolar, compound tubulo-alveolar, or racemose. Some further subdivisions of these are shown in Fig 3.1. Exocrine glands may also be classified on the basis of the nature of their secretions into mucus glands and serous glands. In mucus glands, the secretion contains mucopolysaccharides. The secretion collects in the apical parts of the cells. As a result, nuclei are pushed to the base of the cell, and may be flattened. In classroom slides stained with hematoxylin and eosin, the secretion within mucus cells remains unstained so that they have an empty look. However, the stored secretion can be brightly stained using a special procedure called the periodic acid Schiff (PA) method. Unicellular cells secreting mucus are numerous in the intestines. They are called GOT/FCE/is because of their peculiar shape. The secretions of serous glands are protein in nature. The cytoplasm of these cells is granular and often stains bluish with hematoxylin and eosin. Their nuclei are centrally placed. Some glands contain both serous and mucus elements. Epithelia in secretory portions of glands show specializations of structure depending upon the nature of secretion as follows. 1. Cells that are protein secreting, example hormone-producing cells, have a well-developed rough ER, and a supranuclear Golgi complex. Secretory granules often fill the apical portions of the cells. The staining characters of the granules differ in cells producing different secretions, the cells being described as acidophile, basophile, etc. 2. Mucin-secreting cells have a well-developed rough ER, where the protein component of mucin is synthesized, and a very well-developed Golgi complex, where proteins are glycosylated. 3. Steroid-producing cells are characterized by the presence of extensive smooth endoplasmic reticulum, and prominent mitochondria. In addition to their classification on the basis of structure, and on the basis of the nature of their secretion, exocrine glands can also be classified on the basis of the manner in which their secretions are poured out of the cells. In most exocrine glands, secretions are thrown out of the cells by a process of exocytosis, the cell remaining intact. This manner of secretion is described as merocrine, sometimes also called ecrine or epicrine. In some glands, the apical parts of the cells are shed off to discharge the secretion. This manner of secretion is described as apocrine. An example of apocrine secretion is seen in some atypical sweat glands, and in mammary glands. Finally, in some glands the entire cell disintegrates while discharging its secretion. This manner of discharging secretion is described as holocrine, and is seen typically in sebaceous glands. Depending on the mode of secretion, glands may, therefore, be described as merocrine, apocrine, or holocrine. The secretory elements of exocrine glands are held together by connective tissue, mainly reticular fibers. The glandular tissue is often divisible into lobules separated by connective tissue septa. Aggregations of lobules may form distinct lobes. The connective tissue covering the entire gland forms a capsule/ephyret. When a gland is divided into lobes, the ducts draining it may be intralobular, lying within a lobule, interlobular, lying in the intervals between lobules, or interlobar, lying between adjacent lobes, in increasing order of size. Blood vessels and nerves pass along connective tissue septa to reach the secretory elements. As a rule, exocrine glands have a rich blood supply. Their activity is under nervous or hormonal control. The secretory cells of a gland constitute its parenchyma, while the connective tissue in which the former lie is called the stroma. Endocrine glands are usually arranged in cords or in clumps that are intimately related to a rich network of blood capillaries or of sinusoids. In some cases, for example the thyroid gland, the cells may form rounded follicles. Endocrine cells and their blood vessels are supported by delicate connective tissue, and are usually surrounded by a capsule. Neoplasms can arise from the epithelium lining a gland. A benign growth arising in a gland is an adenoma, and a malignant growth is an actinocarcinoma. In this chapter we have considered the general features of glands. Further details of the structure of exocrine and endocrine glands will be considered while studying individual glands.